Hello, everyone, and welcome to today's GIBCO Galaxy Tour webinar on developing better techniques for 3D culture and analysis. My name is Marie Stone, and I'll be your moderator for today's event. Today's educational seminar is presented by LabRoots and brought to you by GIBCO by Thermo Fisher Scientific. This seminar is part of the GIBCO Galaxy Tour 2021 webinar series designed to help scientists master their cell culture basics and learn more about advanced 3D cell model techniques and applications. There will also be a Q&A session towards the end where our technical experts will address your questions. I am pleased to introduce our two speakers for today. Dr. Edwin Golez, Field Application Scientist of Thermo Fisher Scientific, has over 20 years of experience working in the San Diego biotech and pharma scene as a bench scientist. Leveraging his experience in 3D human tissues for model development and drug toxicity screening, he will kickstart the seminar by sharing about the 3D cell model applications and workflows that Thermo Fisher Scientific has generated in-house. Our second speaker, Dr. Bhaskar Mandavilli, is the Senior Staff Scientist of Thermo Fisher Scientific, who currently oversees the development of fluorescent probes for imaging and flow cytometry applications. With over 15 years of experience specializing in high-content imaging and development of fluorescent reagents across technology platforms, he will be covering the essential tools for labeling and detection in 3D models. Let us welcome Dr. Edwin Golez to start his presentation.
Great, thank you for that introduction. Again, my name is Edwin Golez, the Field Application Scientist here at Thermo Fisher, covering North America. And today we're going to talk about 3D models, developing better techniques for culture and analysis. So here's the agenda for my portion of the talk. First, go into some background on what 3D cell models are. Then we'll go into what is needed to create these 3D models by highlighting the Thermo Fisher 3D cell model portfolio offering. To highlight these products, we're going to go over some 3D cell model applications, some different workflows that we support here at Thermo Fisher. And finally, we'll end with resources and a summary of our talk before we move on to Bhaskar's portion of the presentation. Right, so, why do we want to create 3D models? We want to have an in vitro approach to predict human biology. When we want to predict human biology, we need a way to mimic what we see in the human body by looking at different types of inputs that the body that a body takes in, such as a genetic component, environment, or drugs, and have a way to measure these changes to the inputs by looking at things as symptoms, biomarkers, or outcomes. We create a cell model to hopefully capture that type of complexity, but we'll look at more simplified different inputs such as overexpression of gene or proteins, looking at media systems to mimic the nutrition, as well as the exposure to drugs, and measure comparable outputs to see how those changes in inputs can affect the system. So we're creating cell models to better predict and translate, to predict outputs that best translate to humans. Kind of going into some definitions of what we consider a 3D model. Fatula et al. Has a really nice definition, sort of a textbook definition of what we consider what a 3D cell model is. He describes it as an in vitro cellular cluster from a particular cell source, which is capable of self-renewal and organizing, and has some sort of similarity to the tissue of origin. The graph here on the right here highlights different types of cell models, going from least complex, more simpler models on the left to more complex, more human in vivo-like on the right. Starting on the left here with the monolayer for two-dimensional cultures, it's really your cells grown in a culture dish or flask, usually single cell, a single cell method. It's been used. It's really your drug's first exposure to a living system. So it's been used for several years. It's been can be used to look at like early stage or mechanisms of action, functionality, and also toxicity effects. As we move towards the middle, as we increase complexity, we can add multiple cell types to a 2D model by looking at the co-cultures, which will employ multiple cell types, 2 or more, to see how that interaction better mimics the in vivo system. Another method that's used is the organ-on-a-chip modalities, which utilize a microfluidic device where you have cells placed in these microfluidic channels, and you see the effects that flow and perfusion of media and drug have on the system, something you'd see in the in vivo setting. The monolayer culture I mentioned earlier is usually used with immortalized cell lines, but to add complexity, you can also use primary cells, or cells that are procured freshly from tissues, again, to get that more in vivo-like environment. But what we focus on here at Thermo Fisher, what I support, are the creation of 3D models for better mimicking the in vivo environment. And those can be the self-aggregating models such as spheroids or organoids, or also we can support other types of 3D models such as what you would produce from a bioprinter. Kind of deeper diving into spheroids and organoids, sometimes those we like to kind of clarify between the two. So spheroids, if we go with the Fatula definition, is a cellular cluster, but it's derived from immortalized cell lines or primary cells, which may grow and proliferate depending on the cell type, but they do not change or differentiate into a different cell type at the end of the spheroid protocol. So the cells that you start with at the beginning of the spheroid protocol will be the same type of cells you have at the end. Organoids, on the other hand, will come from a stem cell source, whether it's embryonic stem cells, induced pluripotent, or adult-derived. Again, they grow and proliferate, but they will change, renew, organize into a different cell type or cell types at the end of the protocol. So the cells you start with at the beginning will be different from what you have at the end of the protocol. So why do researchers go into 3D models? This table here kind of goes into the sort of the pros and cons of transitioning from 2D to 3D. I'd say the biggest advantage of going to 3D models is the more representative morphology. And our cells do not grow in a single layer on a plastic dish, lying nicely next to each other. They're grown on top of each other, through each other, with different proteins and components growing through them, which is really more representative of what you'd see in the body. With that proper morphology, the cells are exposed to the nutrients in a more proper way. They're also exposed to the drug, and their sensitivity to the drug is more representative because they're grown in the proper morphology. While those are benefits to having a 3D system, is that more in vivo-like environment, there are certain things you need to consider before going into it. As you know, it's a new system it's a new type of system that people may be going into. So first is that it's a newer model as opposed to 2D models, which have been around for several decades. There's not that many validated or guided workflows for 3D models as there are for 2D models. I think the data is being gathered to generate those, but we're not at that point where we can say this one method is the correct way to do this type of 3D model. They're a bit more complex compared to 2D, more components, more different types of reagents or plastics may have to be used, which also impart a longer time to create the models, going from a magnitude of days for 2D systems to several weeks or months in 3D systems, which can also impart more of a cost in terms of materials, worker time, media growth factors to create a 3D system. So these are some considerations you need to look at before transitioning to a 3D model. But what we like, though, with the 3D models is that they're more physiologically relevant. They provide an access to human tissues, and I think they provide better approximation to what you'll see in the body compared to 2D. So, we get asked, quite a bit, like, what do I need to get started to create 3D models? And we at Thermo Fisher, we have quite a broad offering, I think, that can support you at many parts of the workflow. So the 3D workflow that we supported can support you from creating the model, characterizing, and also imaging and analyzing it. And for the model creation, it can be broken down into 3 more points: your cell source, how you're going to create the model with your matrix or culture vessels, as well as the media supplements as well. And see with the cell portion, we have a variety of primary cells and stem cells to develop different spheroid or organoid cultures. Depending on how you're going to make your model, whether you're going to use a scaffold or extracellular matrix, we have different offerings for that, as well as for non-scaffold-based methods, non-matrix-based, we have specialized plastics to help with your 3D model creation. Also with that, we have different types of media and supplements, many of which you probably use for your 2D culture. They can translate over into your developing 3D model. And you need to verify that the model that you've taken the time and expense to create. Again, they take a bit longer. If you've taken all that time, you want to make sure your model is physiologically relevant. So we have lots of assays and reagents, many of which that Bhaskar will talk about in his portion, to verify that your model is functional and is also healthy in terms of creating a model after all that time. With that, we also have the instrumentation to be able to analyze this characterization, whether it's plate-based, such as our plate-based readers, such as our Veriscan Lux, or our image-based systems, such as our EVOS or our Cell Insight high-content screening platforms. We can also look at gene expression, utilizing our QuantStudio PCR instruments for different types of characterization analysis as well. And finally, another offering that we have here is that we just launched is our 3D cell bioservices, which can be a tool for the lab that may not have the manpower to develop these systems. We'll take that burden off you, and we can do different types of assay development, look at drug screening, and look at your model through multiple assay readouts. So this is really a snapshot of what we support at Thermo Fisher for 3D cell models. And as we go into the next portion, we're going to talk about different workflows and kind of highlight these products in action by talking about particular applications. So the first one I want to highlight here is our neural organoid workflow. You see here we have an application note that is available online, and it goes into creating neural organoids from differentiating induced pluripotent stem cells. So this method here highlights our cell offering, our iPSCs, our matrices, our gel tricks in terms of to differentiate and create the organoids, as well as our media offering to grow and develop it. The bottom graph here walks you through the workflow, starting with your cells in 2D PSC culture. You'll harvest them and place them into ultra-low attachment spheroplates to help form embryo bodies, which are basically PSC spheroids. This is where the clock starts in terms of the timing. We'll have cells form those cell aggregates, and then we'll change the media to an induction media, which allows them to guide them towards the neural pathway. After that, we will go into where sort of the magic happens, where the parroting, where we will encapsulate the induced embryo body with our Geltrex matrix to start the differentiation, spatial organization, and formation of that neural organoid. And then for long-term growth, we will put it into a growth and maturation media, which will allow these organoids to grow for several weeks or even up to months at a time. Here we have some images which are found in the application note of what the cell should look like at different stages. So after the first picture is after the cells are placed in the low attachment plates. They're forming the cell aggregate, as you can see in the middle, and this is before the induction. So you start seeing the cells aggregate, forming a nice PSC spheroid. The middle picture here is at day 7 after they've been in induction media for a bit. You can see sort of a denseness starting in the middle with that defined border on the outside, showing that the cells are primed to go down that neural pathway. The last picture here is after the parroting, the encapsulation. This is more in the long-term growth phase. You are really seeing the the development of the neural organoid with these nice convoluted epithelial structures on the outside, these nice ruffles, as well as the denseness in the middle showing the proper the organization of the organoid. Here's some immunofluorescence pictures highlighting different aspects such as SOX1 neural stem cells here shown in the pink, as well as neurons that are radiating from the outside here on the left is shown in green. So these images highlight after, in this case, 47 days, different types of cell types that are present through our imaging platforms. What I want to highlight here is through our gene expression analysis is that with this method, it really relies just on the aggregation of the cells and the media that we use to form our these neural organoids without having to add in any type of special small molecules or growth factors to guide it down to have it produce a region-specific part of the brain. It's really more of a mini brain, sort of a whole brain snapshot of early developed brain. And the gene expression the gene expression analysis here highlights when we probe for different cell markers, you can see on the right that there's evidence of different cell types from different areas of the brain. So highlighting here on the right, we'll see different types of neurons, evidence of forebrain, neural ectoderm, and also some areas of cerebral cortex and hippocampus. So it's really a nice, I think, a broad snapshot of what we'd see in a early development whole brain utilizing this method. So I think this is a good, this gene analysis kind of shows the range of cell types that are present following this method. Switching gears here from neural organoids, and the next workflow I want to talk about is our cancer spheroid applications. Our R&D team in Bangalore really worked hard to develop these protocols for generating cancer spheroids with these 8 different cancer cell lines. Utilizing 3D models for cancer research, I think, is ideal because you get the proper morphology to mimic the tumor microenvironment and help you understand more so how drugs can penetrate or possibly start developing resistance in a in vitro system. So the team worked for optimizing the protocols for developing spheroids, looked at ways for optimizing the cell culture times and conditions, did some characterization, as well as apply them to different high throughput assays. So it's just a lot of great work from our Bangalore team. So if you click on one of the protocols, in this case, the T47D cancer cell line, this is what you would see. You'd see a nice detailed page first starting on how to maintain the cells in just 2D culture before forming spheroids. A list of the materials that are required with hyperlinks to products that we offer at Thermo Fisher. A nice detailed protocol on cell harvesting, how to plate them, putting them into the Nucleon sphere plates, and also tips and tricks on recommended cell densities and development times, which are catered to each cell type. So each of these protocols will have different parameters based on the cell type that's being analyzed. I think a big help here are some of the images that are shown here, such as this graph here, which highlights different cell densities that we use to form these spheroids, but also looking at them at different days or lengths that they're in culture. So this is a nice visual QC of what your spheroids should look like over time. It's going to make sure that that they're forming properly. I think it's a good way to make sure that you're following the protocol correctly, and I think it just gives you a nice view of what the spheroids on your protocol. And these were bright field images taken with our EVOS microscope. Further characterization that was done were some viability assays, such as our Presto Blue viability for these spheroids, shown here at day 6. As you can see, at 6 days with increasing cell density, as what you'd expect, you'd want to see more higher cell viability, which is indicative by the graph. While this assay, I think, shows relative viability, it doesn't give you a sense of the number of dead cells that are present. So to assess more detail of the viability, they performed live dead cell assays to highlight the number of live cells, which are highlighted in green, but also the presence of necrotic cells or dead cells that are formed in the middle that you'll see with increasing cell density. So I think that these images kind of give you a nice, along with the bright field images, kind of give you an idea of what the cell should look like, but also the relative health of the cells based on your length in culture and also your seeding density. So it gives you some options depending on your experimental parameters of how long you should have your protocol run for. The next workflow I want to highlight here is our hepatic spheroids, which can be used for looking at metabolic assays. What's nice about the hepatic spheroids is that the first two applications I talked about are comprised of a bunch of parts that you put together to create that workflow. This is actually the first one of our first 3D product, which is actually sold as a kit itself. So under one catalog number, it'll provide you all the materials that you need to form this model. Utilizing hepatic spheroids versus 2D has, I think, definite advantages. Your cells are in culture longer, going from just days to a matter of weeks. Higher level of CYP activity to measure drug response, but also better gene and protein expression. So within the kit, it has everything that you need to create your model, your cell source, the primary hepatocytes, the plates to create the spheroids, the nucleon spheroplates, as well as the media required for growth and development. And the images here on the right highlight what the spheroids should look like following the protocol, as well as the increased CYP activity comparing 2D to 3D. You can see the advantages of 3D. If you carry it out to 3 weeks, you have the same amount of CYP activity as you would see in 2D after only 5 days. So you're really lengthening your window of being able to use the cell model. This slide here really highlights the simplicity of the. Let's say you start you start the experiment on Monday. You start by thawing, counting, and plating your cells, and you plate them in the nucleon sphere ultra-low attachment plate to form the spheroids. Put them in the incubator. You don't have to touch them until Friday, where you do a media change and let them go over the weekend, and then on Monday you could start your different assays you want on the formed spheroids. So really, I think it's a very simple set it and forget it workflow, which allows you to get within about 7 days workable hepatic spheroids to look at drug metabolism. The last workflow I want to highlight is our utilizing another plastics product, our cell culture inserts and carrier plate systems to develop 3D human skin. A little bit on the cell culture inserts here is that that they come in 3 different well formats: 6, 12, and 24. They have the insert is a polycarbonate semi-permeable membrane of different pore sizes. And the uniqueness of that is it has an adjustable height feature, which allows you to place the insert at different heights within the well, which is highlighted here in the image below. The carrier plate function is nice because for the multi-insert plates, you're able to take all the wells out all at once, and you're able to exchange media or collect media easier because you can take all the inserts out all at once. And these plates can be used for different assays: transport, co-culture, invasion, as well as chemotaxis assays. So they're very useful plates. So the skin model system has an application note. We'll just walk through a little bit the protocol to generate it. You start by coating the insert with some protein coating matrix and ECM for about 30 minutes. And then you're going to seed adult keratinocytes on top and let them sit submerged, with the cells submerged underneath the level of the media for 2 days to let the cells set onto the insert. After 2 days, you're going to bring the media level down to the air-liquid interface, or ALI level, which is when you put the media just at the bottom, to the level of the insert bottom. So the bottom of the cells will be in contact with the media, but the cells on top will be exposed to air, allowing the keratinocytes to stratify and differentiate into the different layers of skin that you would see. You let that go for 12 days, feeding accordingly, and then allow for your cell health and characterization to be formed after that. So I think to what's great about the interest again is was the adjustable height feature, and in this protocol, which that adjustable height feature comes to play and allows you to have flexibility in terms of your feeding schedule. For example, if you were to place the insert on the high level, which would be the level here on the far left of the graph, to get to ALI, you would need 1.5 mils of media in the well. But if you have at the high, you have more media in the well, you don't have to do any type of feed for 4 days. So you can adjust the level of when you have to do any type of cell maintenance based on the height of the insert that you placed it within the carrier plate. So allows you some flexibility in terms of how you do your protocol, allow you to maybe not have to come in on a weekend or during a busy time because of this the adjustable height feature. And this picture is shown in the app note here, and we have, as you can see, if you take a cross section of the skin, you can see the different layers of different skin layers that have been differentiated, and you could probe for these by looking at immunofluorescence or immunohistochemistry or gene expression for these different types of cell markers, the cytokeratins, loricrin, or involucrin, just to show different ways of highlighting the different layers that have been produced. You could also look at cell health by looking at live dead cell stains, and these all could be imaged utilizing our EVOS imaging systems or gene expression through our Quant Studio real-time PCR systems. Finally, if we go looking at some resources that we have for 3D, if you go to our website, thermofisher.com/3D models, the website here can kind of guide you along your 3D cell culture journey. Here we have many key protocols and methods and relevant products, many of which I've highlighted here in this talk. The application notes for the different workflows that we talk about can be found here. We have scientific posters that are available, kind of showing the latest and greatest which is going on in our R&D group. Another way of showing which highlight our products in action, and are also been presented at different scientific conferences around the globe. Other educational resources, such as our 3D cell culture handbook, a very comprehensive handbook on different culturing techniques, methodology, characterization, instrumentation. I think it's a good resource for either the beginner who's looking to transition from 2D to 3D, or the person that's more experienced, which is knows how to create a model, is looking for different ways to improve on what they've already used. There are digital training and e-learning courses that are available, such as how to create neural organoids, getting started in 3D cell culture, and one that we just produced earlier this year is on 3D cell culture and analysis. So kind of walking you through, the training is going to walk you through different parts of the 3D cell culture workflow. And finally, we also have the Molecular Probes Handbook, which goes into all the different fluorescent labeling technologies to highlight the cells, which can be a key aspect when you're trying to develop your 3D models, being able to image and analyze your complex model. So finally, in summary, I hope we've been able to convey that 3D cell culture models better recapitulate the in vivo environment, but in an in vitro way. Here at Thermo Fisher, we can support the entire 3D cell model workflow, going from model creation to characterization, and as well as to imaging and analysis. We've optimized protocols for different workflows, such as neural organoids, 3D skin, cancer spheroids, as well as hepatic spheroids. And finally, visit our website at thermofisher.com/3D models for useful information on how to create and analyze your desired 3D model. Thank you.
Thank you, Edwin, for the insightful session on 3D model workflow and applications. I am sure that your audience will be interested to visit the educational resources online for more information. Next, I would like to invite Bhaskar to share more about the essential tools for labeling and detection in 3D models.
Thanks, Edwin, for a great introduction on how to grow spheroids and then all the complexities and tricks to grow both spheroids and organoids with different models. So today, my name is Bhaskar Mandavilli. I am a senior R&D manager in molecular probes division of the Thermo Fisher Scientific. I am involved. I manage a team involved in developing fluorescent reagents that can be used to measure a lot of different functions in both monolayer cells and 3D spheroids and also tissues. So when Edwin already touched upon the cell culture part, I'm not going to talk too much about that. But when you look at the steps to label and detection of 3D models, it is similar to what we do with 2D models. The only difference is more complexity with the 3D models in terms of staining and also analyzing the samples. So the step one, Edwin already talked about, and then step two is about labeling and optimization, which I'm going to give you a few examples in today's seminar. And then step three is in imaging analysis, and I'm going to talk to you about two different platforms using a wide field microscope to just check the spheroids and the staining part, and then using a little bit higher end microscopes after that, like confocal microscope or CX7 high content imaging platforms. So when you would look at labeling and optimization, molecular probes we have about three different strategies in this space. One is conjugated antibodies. Both you can conjugate primary antibodies using our conjugation kits, but also we have several Elixaflor-based conjugated IgGs that can be used as secondary antibodies to stain your samples or stain your markers. And then we have fluorescent proteins based on baculovirus system, where you can mark your cells when you are growing spheroids. For example, if you are growing heterogeneous cell populations in spheroids or organoids, you can mark them using either GFP or RFP using our BACMEM-based fluorescent proteins. And these are very useful for both live or fixed samples, because these are retained even after fixing the samples. So you can look at the demarcation of different populations within the spheroid or organoid after fixing your samples, and also after staining with your antibodies of interest as well. And then we have organic fluorophores, which are small molecules. And these are mostly used in live cells, live spheroids or organoids, and also some of these probes can be used in fixed samples as well. And you can also fix after staining the spheroids or organoids. So when we talk about labeling and optimization, there are some minor differences between when you use 2D monolayer protocols compared to the 3D samples. Most of the times, you use the same concentration of the dye that you use for 2D monolayer cells, but most of the differences come when you are doing the treatment with the drugs or staining time. That the incubation time when we are, it also depends on each fluorophore. Some fluorophores are much better in diffusing into the and staining interior into the spheroid or organoid, while some take more time. It depends on what dye you are using and what function you are measuring. And also, it is a little bit more complicated when we are trying to do antibody stainings, because they are a little bit bigger molecules. You may have to use more tricks to get the antibody into the interior of the spheroid or organoid. So when you are talking about a cell health continuum, either in a tissue or a 3D organoid sample, we are talking about, when we are measuring, we are talking about looking at healthy cells, what the phenotype looks like, and also looking at stressed populations and apoptotic cells and dead cells. And also sometimes there is inflammatory response when you are talking about especially tissue or spheroids or organoids after the dead cell after some of the dead cells are dying in the population. Yeah, when we are talking about cell health continuum, as you can see here, for each state of the cell in the population, you can measure different parameters. For example, for a healthy cell, you can look at mitochondrial membrane potential. You can look at the cell proliferation. As we all know that there is a doubling time of each kind of cell lines. And also you can look at morphology, which is mostly intact in a healthy cell. So when cells are stressed out, either due to tissue environment or due to drug treatment that you do into the cells, the stress can be either undergo recovery into the healthy cell, or it can go to the other side where the cells are dying, whether due to apoptosis or necrosis, which can be measured by either caspase activation, DNA damage, or loss of membrane permeability in the cells. And also, as I talked to you before, after the cells are dying, there is inflammatory response or phagocytosis that happens in the tissue environment and also in spheroids and organoids. So now I'm going to talk about a little bit about mitochondrial depolarization and oxidative stress measurements as a result of stress. Here is an example where this is a monolayer population. Just I want to talk about how these reagents work. Here is an example where we used TMRM, which measures mitochondrial membrane potential, which is high in healthy cells. There is more staining in healthy cells, and then we measure apoptosis using oxidative stress using CellRox green reagent, which is a fluorogenic reagent to measure oxidative stress in cells. It is dark in healthy cells when there is not much oxidative stress, and when you treat the drugs with an oxidant, you can see increase in staining of CellRox green reagent and decrease in TMRM mitochondrial membrane potential indicator, which says that as you, with time after treating with the drug, there is reduction in mitochondrial membrane potential as the cells are stressed out and increase in oxidative stress within the cells. These are U2OS cells stained with 50 micromolar menadion. And here is another example where you can measure these two phenomena or markers in a spheroid sample. Here is an example where we did it on confocal microscope. This is A549 3D spheroid models where we stained with nuke blue live ready probes reagent and then MitoTracker orange, which again like TMRM measures mitochondrial membrane potential, and then CellRox deep red reagent. This is a healthy spheroid. This is not a treated spheroid. This is an example just to show that you can measure across the Z in a spheroid using confocal microscope. As you can see here, you can count the cells using nuke blue live ready probes reagent, and then you can also look at the MitoTracker staining an entire spheroid, which shows the healthy cells, and there are some sparsely populated cells that stain for CellRox deep red reagent. CellRox deep red reagent, similar to CellRox green reagent, measures oxidative stress in cells, but in a different spectral profile. And when it comes to staining, both MitoTracker orange and nuke blue live reagents are very good in penetrating the spheroids, and we do it for 30 minutes. And when it comes to CellRox deep red reagent, it is not as permeant in the spheroids, so we use a little bit more like a 2-hour staining for this CellRox deep red reagent to penetrate into the spheroid and stain all the cells in the spheroid. So next one in the cell health continuum is measuring apoptosis and DNA damage, which is once the cells pass through the stress response and they can no longer recover and they are dying, you can measure those using again TMRM mitochondrial membrane potential indicator and solvent caspase 3/7 substrate, which is cleaved in only in apoptotic cells. You don't see much staining in healthy cells. As you can see, this is a population of HeLa cells, monolayer cells, where where we did treatment for 6 hours using 500 nanomolar staurosporin. As you can see, over time there is a decrease in mitochondrial membrane potential, as seen by decrease in TMRM staining and increase in solvent staining to mark the apoptotic cells in the population. Again, here the staining with these reagents, both TMRM and solvent caspase reagents, are very penetrant into the spheroids. I will show you the next one, spheroid population. Here's an example where we stained A549 cells after treating them with different doses of niclosamide, which is a drug that induces apoptosis and also causes depolarization of mitochondrial membrane potential. Here the rows show the multiples. Here the n is equal to 6, and then the columns show increasing concentrations of niclosamide drug. As we increase the concentration of the niclosamide, as you can see in this left side in the image, you can see that there is decrease in mitochondrial membrane potential and increase in apoptotic cells. And this can be easily quantitated using our Cell Insight CX7 laser illumination based high content screening platform. You can measure the number of cells that stain with mitochondrial membrane potential, and also stain the population, and then quantitate the population that stain with solvent caspase reagent as well to mark the apoptotic cells in the spheroids. As you can see here, with increasing concentration of niclosamide, you can see decrease in population that stain with mitotracker orange, which says that with increasing concentration of niclosamide, the cells are losing mitochondrial membrane potential, and there are more apoptotic cells as seen by staining with the caspase 3/7 reagent. Here is another example where we show the T cells attacking a spheroid and killing the cells in a SKBR3 spheroid. And then again here the cell death can be seen by solvent caspase 3/7 reagent, while the T cells are stained with cell tracker deep red. Because these are stained separately, the staining is for 30 minutes with the cell tracker deep red reagent, while in spheroids we stain after the T cell penetration, and for 24 hours we stain it with solvent caspase 3/7 green reagent for about 30 minutes. And this is an example where spheroids are stained with a hypoxia reagent. This hypoxia reagent gets fluorescent only when the percentage of oxygen goes down below 5% in cells. As you can see here, there are some cells which have brighter staining compared to other cells based on the level of hypoxia in the cells. And again, I want to point out that there is not like a very good gradient of hypoxia in spheroids as compared to a cancer tissue, because there are no blood vessels in the spheroids, right? So it depends on more like diffusion of CO2 and oxygen into the spheroids rather than a very perfect gradient that you usually see in a cancer tissue. And the next set of reagents that I'm going to talk about is a reagent that measure the plasma membrane integrity in the dead cells. This can be both necrotic cells or late apoptotic cells. As we know that during apoptosis, it undergoes DNA damage, nuclear condensation, and caspase activation that are earlier parameters. But in the end, these cells lose plasma membrane permeability as well, and then they dissociate and then engulf during phagocytosis process. And then there is also another way of cell death, which is necrosis, which is more drastic and leads to loss of plasma membrane integrity, leading to cell death. Here is an example where there is antibody-mediated cytotoxicity. These are SKBR3 spheroids. As you can see here, where the natural killer cells are added to the SKBR3 spheroids, and then the NK cells are stained with, as described there, cell trace, violet reagent for 30 minutes, wash the reagent off, and then added to the spheroids, left over 6 hours. And then we measure dead cells using live dead kit, and the green staining is calcine, which signifies the live cells, and then as you can see, the red dead cells are stained with the ethidium homodimer stain that comes with the live dead cell imaging kit. As you can see, the dead cells are marked with the red staining in these spheroids. This is again earlier time point in the treatment with NK cells, so you don't see many dead cells in these spheroids. Here is an example where we have grown spheroid A549 spheroids and then added NK cells with or without cetuximab drug. Cetuximab drug is a anti-EGFR drug, which actually kills the cancer cells by inhibiting the proliferation of specifically cancer cells. As you can see here, minus cetuximab and minus NK cells, you see a little bit of apoptotic cell population, and when you add NK cells in 6 hours, you see a little bit more increase in apoptotic cell population as measured by cell event caspase reagent. And then when you add both cetuximab and NK cells, there is potentiation of killing of cells mediated through NK cells, and you can see increase in apoptotic cell population within the spheroid. And you can also see here the NK cells penetrating the spheroid and killing the cells by apoptosis as well. Here, using our Cell Insight CX7 laser high content screening platform, you can see you can actually quantitate these apoptotic cell populations very easily. And as you can see here, as shown in the previous slide, you can see increase in apoptotic cell population as we go from no NK cells to plus NK cells, and also when we use antibody plus NK cells, there is even more increase in apoptotic cell population as measured by caspase reagent. Again, the last step in labeling and detection is using our microscopes and then some other tricks to efficiently measure these spheroids. Here is an example using a wide field microscope. When you use a wide field microscope, the light is dispersed, so you don't see very clear delineation of cells, especially when you use thick tissue samples or spheroids or organoids. And when you use confocal microscope, as you can see here, there is more pointed illumination of light of the sample, so you see more clear delineation, as you can see from previous slide where there is wide field microscopy. Here, using confocal microscope, you can see more clear delineation of the staining in spheroid sample. This is an example of hepatocyte spheroid measured on our Cell Insight CX7 laser system using laser illumination. And also another way of improving your resolution within the spheroid or organoid is clearing reagent. So because most of the times we use the media or PBS in the spheroid or organoid sample, so there is some differences in refractive index between the objective of the microscope that you use and the sample which is in aqueous phase. So to reduce, to match a little bit more of the refractive index between the objective and your sample, you can use one of our Cytovista clearing reagents. So this is basically diffusing into the sample and matching the refractive index. So there is more penetration of light and then more resolution, into the spheroid sample. As you can see here, after clearing with Cytovista clearing reagent, you can see, you can measure, you can see more cells, and also you can measure more, quantitate more cells within the spheroid. Here is another example where the spheroids of different sizes are grown. As you can see here, the spheroid size can go anywhere from 50 microns to about 300 microns with increasing number of cells when you grow spheroids. And they were stained with Cytox green, nuclear dye, and then as you can see, before clearing, as increase the size of the spheroid, you cannot count all the cells within the spheroid, and after clearing you can see there is increasingly clear delineation of the cells, and also you can count those cells using high content imaging systems. So you can use our EVOS M7000 imaging system in an automated way, both in 384 well plates and 96 well plates. This is a way of looking at your spheroids, quickly looking at your spheroids and also your staining patterns using this. But again, as explained before, this is a wide field imaging system, so you don't get clear delineation of sample within the spheroid. So this is just for checking your sample. And as you can see here, you can look at all the spheroids and this is a 384 well plate scanned. It takes about 3 to 4 minutes to scan through the do a quick scan and look at, okay, there are spheroids in all the wells and they're looking good. So you go to the next step of going to a much higher end high content screening platform, which has a laser illumination and also confocal microscopy system. And also it comes with a smart scanning. We call it Eureka scan, where you can quickly scan through a plate and delineate your spheroids. Because especially when you're growing in a 96 well plate or 384 well plate, the spheroids can be at different places within the 96 well. So using a smart scan or Eureka scan will allow you to quickly scan at lower magnification and identify your spheroids. And when you go to higher magnifications in the second round, you can actually just go to the spheroids and then image them instead of going through all the fields, which takes about a lot of time. Here is an example where you identify using the Herc staining. You identify, here you can use even bright field or Herc staining at a lower magnification. You can use 2x or 4x, quickly scan through the plate, and then you go to, on the bottom panel, you can see, you can go to higher magnification like 20x or even higher magnifications, and then just go to the sample and scan through it. Here is another example of identifying sample at lower magnification and going there to the particular fields. It will save a lot of time. If you don't use Eureka scan, it may take up to 3 to 4 hours to scan through a 96 well plate. And then when you use Eureka scan, as you can see here, it takes about 28 minutes, 24 seconds to go through a 96 well plate, because both first round of scanning and second round of scanning, because you don't have to scan through the entire field of view. You just go through the specific fields that has spheroids, and then you scan through at higher magnifications. So I will stop here, and then thank you for your attention, and I am ready to take questions if you have any.
Thank you so much, Bhaskar and Edwin, for sharing your expert insights into these topics. We'll now start the Q&A portion of the webinar. If you have a question you'd like to ask, please do so by clicking on the Ask a Question box located on the far left of your screen. We will do our best to address as many questions as possible. In the event that we are unable to do so, representatives from Thermo Fisher Scientific will reach out via email with an answer. It looks like we have got a few questions already coming in. Perhaps we can get started with questions for Edwin. So the first question for Edwin is, can you summarize the difference between Geltrex matrix and other ECM? And what kind of organoid culture applications would you recommend for the use of Geltrex?
Yeah, thank you. That's a great question. Geltrex is one type of what we call a naturally derived ECM, which is in this case, a reduced growth factor basal membrane extract that comes from a mouse tumor. Within this matrix, it has, the different types of components, collagens, laminins, intactins, and other components that comprise this extracellular matrix. It also helps, the Geltrex also helps form an organized scaffold to provide structural support for your tissue, as well as offer functional input that will help modulate cellular function, helps with cell-to-cell communication and growth and development. Geltrex also is being one of the natural ones, is also a temperature-controlled material, which in this case, when you warm it up, it'll actually polymerize to create the solid material. So as a natural ECM, other examples of this would be collagen, laminin, and gelatin. Other types of ECMs that are available are synthetic ECMs, which actually rely on synthetic polymers to create the stiffness. So by looking at the physical, biological, mechanical properties of these scaffolds, you can tune them independently by changing molecular weight, cross-linker, and using a different type of way to polymerize. So what's nice about the synthetic one is you have control over how stiff you make your extracellular matrix, and also how to time when it happens. And examples of different types of synthetic matrices are polyethylene glycol, as well as polyglycolic acid-based ECMs. To the second part of the question, for what kind of applications would you recommend for Geltrex, I'd say you can use Geltrex for different iPSC-derived organoid models, such as neural organoids, which I highlighted here, patient-derived organoid models, such as intestine or cancer-derived patient sample organoids. They can also be used to develop spheroid models by adding them in with the cells to help the cells aggregate and form nice compact spheroids, which is highlighted in the cancer spheroid protocols that we discussed.
Okay, thank you. Next question. What are the pros and cons of using low cell attachment plates versus non-treated polystyrene plates for the 3D spheroid culture? What are some key considerations to help me, a researcher, decide between the two options?
Yeah, another great question. Both of these types of plates have been used to generate spheroids. With the non-treated one, polystyrene ones, I think it's more of an older method. The non-treated plates, we'll start with them while they have a hydrophobic surface, similar to what you see with a ultra-low coated attachment plate. The non-treated plates are mainly just for cells growing in suspensions that actually do not really need that. The endpoint is not to actually have to have them aggregate, but just rather not have them attached to the surface. Again, this is an older method, and they were used early on when, I guess, spheroid forming methods were used, such as the hanging drop method. But for easier and more consistent cell aggregation, or to create 3D models, I recommend those low attachment plates, such as our Nuncon spheroid plates. Since you're trying to get the cells to aggregate rather than not lay down as a monolayer, these low attachment plates are the way to go. They promote uniform cell aggregation to generate reproducible spheroids, and minimizing that variability in a developing 3D model is key, as the OLA plates are designed for that type of that formation.
Okay, thank you. And the last question for Edwin, can you share more about the kind of controls you have in place or parameters you are measuring to ensure the reproducibility of the neural organoids generated in the lab?
Yeah, I think this question has been asked in the field as well too. I think in general, I think I'd recommend that depending on the model, you develop some general QC parameters to help with those types of consistency issues. Like, for example, looking at the size of the model, appearance, like gross morphology through immunohistochemistry or fluorescence staining to look at spatial organization. Another thing you can look at is doing a small qPCR panel to make sure that the cells are differentiating, proceeding into the new cell types that should be forming over time. So just little ways to make sure that the model is progressing in different ways. Specifically, say for the neural organoids that we're talking about here, certain things that you would look for would be epithelial outgrowth during the expansion phase. So you want to look for an increase in size and a change in morphology, and this stuff should occur approximately at the same time. If you were to do this experiment several times, the timing of these changes should happen at the exact timing to make sure that the organoid is developing properly. So those are things that I'd recommend. But also depending on the model you're generating, whether it's whole brain or region specific, some of these recommendations maybe have to be tailored to that model. So specific recommendations may be difficult to make, but those are just a couple of things that I would suggest.
Great, thank you. Thank you, Edwin, for answering these questions. And now we'll proceed with questions for Bhaskar. The first question is, I would like to confocal image my spheroids with minimal distortion to their 3D structure and with as little sample drift as possible. Can this be achieved with glass bottom microplate or microwell? If so, what are some tips and tricks to get good images for analysis?
Yes, especially use confocal microscope.
Yes, so I would like to confocal image my spheroids with minimal distortion.
Yeah, it is a great question. And then when we try to image this on confocal microscope, I would suggest you use as much as the thin glass bottom plates to image these, and there is always some drift because when we are, especially when we are putting it in the media, I would suggest that you either use a clearing reagent which puts the sample in a place, or probably mount it in one of our prolonged gold mountains, which will help you to keep at one place so that there is not much drift when you are actually, especially doing the Z-sectioning of the spheroids or organoids. And that should help with the drift. And if it is a fixed sample, usually you don't see too much drift. You can actually do without mounting, and then there is always you can use other methods of clearing to reduce the diffraction of the light so that you can get more penetration into the sample, but otherwise it should be fine.
Thank you. And our last question for Bhaskar, for researchers beginning their journey in 3D spheroid culture, can you advise on the types of microscopes and additional technologies that will be needed versus 2D to observe their 3D cultures?
Yeah, in my experience, you don't need entirely different setup for 3D spheroids, but the only thing is there are some minor differences, like using U-bottom plates for growing spheroids, and sometimes you may have to use ECM and Geltrex and other methods to grow spheroids or organoids. When it comes to imaging itself, if you have a confocal microscope or a wide field microscope in the lab, that should cater to your needs. But if you want to do quantitation, probably you may need something like our Cell Insight CX7 imaging system, where you have both wide field and confocal microscopy in there, so that you can not only image but also quantitate your sample very efficiently on the fly.
All right. Thank you, Bhaskar, for your replies. And to our audience, I hope the sharing has been helpful to your current research work and future plans. This wraps up our GiveCo Galaxy Tour webinar on developing better techniques for 3D culture and analysis. Once again, I would like to thank Edwin and Bhaskar for sharing with us their knowledge, application notes, and essential tools for developing and analyzing 3D cell models that could enable better understanding of complex biology in a physiologically relevant context. A big thank you to our audience for joining us today and submitting interesting questions. For those questions that we were unable to answer live, we will follow up with an answer via email. We hope all of you have gained more knowledge today that will help you to enhance your lab skills. We would also like to thank LabRoots for underwriting today's educational webcast. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Thank you, and stay tuned to more educational webinars from Thermo Fisher Scientific.